was inducted in Indian Academy of Sciences in 2024. So, my research group, we work in the field of biological inorganic chemistry, and since it's a very diverse audience that we have today, so I would like to keep it the way so that it's understandable to large audience. So to start with, I will be talking about the redox-active iron complexes, but before that, let's give a brief overview of what we do in the research group. So our research group, it's in the field of biological inorganic chemistry. We work with the biologically relevant ligands, and the ligands which are of our relevance are the one which coordinate to the transition metals in the redox intracellular environment. So this could not only affect the on-target efficacy, but also introduce new reactivity. For instance, redox chemistry of some of the transition metal complexes which are formed in situ. So once we make these ligands, their complexes, we utilize them for malignancy, cancer, and also at times for infectious diseases. Of late, we have started gaining lot of interest also towards neuro disorder, especially Lafora disease with the biological science department at IIT Kanpur. Now, as I said that we start with the synthesis of ligand in our lab, we make their metal complexes. At times, we also do their nanoaggregation or nanoformulation. Once we have done that, we try to do the characterization in the in vitro setup, where we see what is the identity of the compound, what's the characterization in terms of what's the binding mode, what's the oxidation state, and all those properties of the metal complexes. Then we go for the in-cell study, where we establish their mechanistic profile, their biological profile, that if they are killing the cancer cell, what is specific cell death pathway they are following. And none of this would make sense if we do not show the translational aspect of our work. When I translational aspect, we mainly work with the animal models, especially so far we with the mice and rat models, and all of these experimental data are further validated by theoretical aspect. So of all the projects, as I said, we are working in our lab. I will be mainly talking about one of the major projects which has been recently published. So to start with, cancer, I will not go into the history of cancer. By now, I think most of us are already aware of different cancer treatments which are going on around the world. And of all the cancer treatment, immunotherapy has made a major stride, to the extent that it's also said that in future, many surgeries will eventually be replaced by immunotherapy. So immunotherapy is playing a very significant role. Now what is immunotherapy? So when we talk about immunotherapy, here our own body's immune system, it is used to combat the cancer cell. Now when it comes to immunotherapy, it's not always, the results are not always very positive. So it varies among the patients even with the same cancer type. If I talk about mouth cancer, if I talk about colon cancer, even with the same cancer type, their feedback or their response or the efficacy of immunotherapy, it varies from patients to patients. And why is this so? Because of the immune characteristic of the tumor. Now, despite several transformative impact of immunotherapy over the period of time, very minute number of patients, they show positive response to the immunotherapy. And that's why this concept of the cold and the hot tumor was proposed, because it talks about the immune status of the tumor. So in order to predict what will be the tumor response, what will be the treatment results, it's very important to understand the difference between the cold and the hot tumor, what will be the intrinsic and extrinsic factors responsible, tumor microenvironment, and also tumor characteristics. So let's start with the cold and the hot tumors. So what are cold tumors? So these are the class of tumors which are poorly infiltrated with the immune cells, especially T cells. You must have heard about the terminology T cells in the immune cells. So in this case, it becomes very difficult for these, since there is hardly any T cells. So immune response is kind of inactive. At the same time, if we talk about the hot tumors, they refer to the they do show signs of inflammation. There is lot of infiltration of the T cells, and they are always ready to fight the cancer cells. Now, why these cold tumors? They evade the immune surveillance. What's the reason? And the reason is that they create a very immunosuppressive environment around themselves, and that's why they become resistant to the immune therapy, immune-based therapy. So that's why, in the recent time, a lot of research is going on to convert these cold tumors into hot tumors, so that the tumor treatment, they are responsive to the immune-based therapies. So all the scientists, many scientists, they are interested to understand if and how we can switch these cold tumors into the hot tumors. And the research obviously goes towards reversing the suppressive microenvironment which surrounds these cold tumors. Now, suppose even if I convert this cold tumor into hot tumors, how do I prove that? How do I verify that? So there are three major hallmarks which are present in the cells in general, which act as a marker for these immunogenic cell death induction. The very first one, I would say, is HMGB, which is one of the nuclear protein which is released from the nucleus to the cytosol, and it's also actually called as the danger or the alarm based protein. So these proteins, they alarm the immune system about the damaged cell. The second one is the ATP release, which is also called as the find me. So it's released outside the cell. This is the second marker for immunogenic cell death, and it helps to find the damaged cell. The last one is the skeletal reticulin translocation. So these are certain proteins which are present on the surface of endoplasmic reticulum. So these are all different organelles which are present inside the cells, and together these three markers, they are known as the damage associated molecular patterns. So these are the three major markers. Other than that, people who work in the field of molecular biology, they have found different markers other than these, but these are the three major markers. So if suppose today I convert the cold tumor into hot tumor, these are three markers which I will be looking for mainly. Now, despite all these advances, there are two significant challenges which is associated with the cancer therapy. The very first one, especially with the cold tumors, it's difficult for these drugs to reach some of these deeply seated tumors. And why is that so? Because there is very minimal vascularization, or if I say the blood supply or the blood flow, it's really low. So that's why these drugs, they cannot reach these deeply seated tissues or the cancer areas. Second one is the difficulty in the real-time monitoring of the treatment, particularly in the near IR range. So there is no non-invasive method to find how the treatment is going. Okay, if the drug is working, if the tumor is going down, if it's decreasing. So there is non-invasive way, there is lack of these non-invasive way. So that's when our role came in. We wanted to show the convergence of therapeutic and diagnostic. So we wanted to synthesize a class of compounds which can not only have a therapeutic potential against these cancer cells, at the same time they can help in the diagnosis or especially in the near IR region. So there is actually a very significance of when I am saying this near IR2 region, because if we go further down towards near IR regions, that's where we get a lot of background signals from other hemoglobin, myoglobins. So that's why it's preferred to have in the near IR2 region. So our research group, we have been working on histone deacetylases proteins and their modulators. What are histone deacetylase proteins? So based on the cofactor requirement, structure, subcellular localization, they can be divided into 4 different classes. So these are the proteins which are present inside the nucleus, and in short I would say they are important for the survival of cells because they participate in the DNA synthesis eventually. So these certain proteins, if you see over here, these are the NAD plus dependent histone deacetylase present in the mammalian cells, and we are mainly interested in these certain class of the proteins. So in our lab we started making different analogs of these inhibitors or the modulators of certain proteins. So we made approximately 80 different library of these certain protein modulators in our, so we have a synthesis lab, and we started exploring these class of compounds by making, especially if you see this oxygen, nitrogen and oxygen binding moiety. So it shows that they have property to coordinate to the transition metals. So once we made these library of compound, we coordinated them with different transition metal complexes, and here is the one which is one of the most active compound which was found. So we coordinated with iron, Once we had these active compounds, we characterize them by different spectroscopic methods like crystal structure, EPR spectroscopy, Moss-Bayer, UV-Vis binding. It's very important before you move on to do any biological assay, the compounds that you are making in lab should be pure enough. And in case if there is an impurity, you will not know that the activity that you are actually getting, it's coming from the compounds that you have synthesized or any leftover or any decomposition product which is in there. So these spectroscopic characterizations are very important. So, but before we moved on to do any biological assay, we wanted to characterize that what is the identity of these in the solution state, because we had the crystal structure, but we were not sure how is it existing in the solution. Now this is one of the media which is used to grow the cancer cell, especially most of the breast cancer cells, then colon cancer cells. So this is one of the media, and what was seen that our class of compounds, they are existing in the form of a nanoaggregate. So if you see this DLS data, it shows that it is existing in the form of a nanoaggregate. Now there is significance that why we want these compounds to be in the form of nanoaggregate. So if you remember, one of the things that I mentioned, that it's very one of the limitation of these class of compound is that their uptake is not that great. So these nanoaggregate, it actually helps in the uptake more than the class of the compounds which were not forming these nanoaggregates. Once we had the characterization of these compounds, we moved ahead to do this cytotoxicity and the cell-based cell death mechanism. So we did this study in 4 different cancer cell line, MCF7 which is breast cancer cell line, A549 which is lung cancer cell line, this is prostate cancer cell line, then this is more aggressive form of the cancer cell line, and this is healthy, just to make sure that our compounds, they are not acting as a poison. So you have to make sure that you also treat your compounds towards the healthy cells to see that they should not be toxic towards your healthy cell lines. So, and what was found that our FeL1 was found to be most active among all the other compounds that were synthesized. So here are some of them. Once we got the activity in the cancer cell line, we moved, so the story does not stop here. Once you synthesize the compound, once you screen them against different cancer cell line, that's when the actual story starts, because you don't know what mechanism of the cell death pathway. It's very much like this, if you are eating an antibiotic, eventually you have to know where is that antibiotic going, where is it targeting, what cell death pathway. So that's when the actual story starts. So we started with the cell death pathway study, and this study is done in the presence of different inhibitors. So we had inhibitors for the apoptosis, necroptosis, ferroptosis, paroptosis, autophagy. There are more cell death pathways, but these were the initial that we did. And what was found that our class of compounds, it is undergoing autophagy mediated apoptotic cell death pathway. Now once we have proved in the presence of inhibitor, we wanted to further verify it by immunofluorescence assay, and what was used was LC3 puncta, which is one of the autophagy marker. So we saw these LC3 puncta coming on the surface of these autophagosome in the form of this green fluorescence which we see over here. So this is the immunofluorescence assay to further verify that that is the cell death pathway that they are undergoing. Now during this process of autophagy mediated cell death pathway, these class of compounds, they are iron complexes, so they are redox active. If they are redox active, they can go from iron 2 to iron 3, iron 3 to iron 2. In the process, they generate reactive oxygen species. So you must have heard about all these anti-aging drugs. Okay, so those are some of them, or most of them actually, they are quencher for these reactive oxygen species. So it means that all the time in our body, a lot of ROS is being generated. And so this class of compounds also, it generates ROS, and that's one of the cell death pathway, you know, like it goes inside the cancer cell, participate in the ROS generation. This reactive oxygen species goes and targets different organelles, and one of them is it causes the glutathione depletion. At the same time, one of the protein which uses this glutathione, so glutathione is also one of the millimolar concentration antioxidant present naturally in our body. So the glutathione depletion is taking place. This is one of the substrate for the glutathione peroxidase protein, which prevents the lipid peroxidation inside the cells. So if this is getting depleted, glutathione peroxidase will get depleted and lipid peroxidation is happening. So these are assays. I understand it's a lot of technical things over here, lot of dyes and all, but the overall concept what is we are seeing over here, that is participating in the ROS generation, and at the same time the other ingredients inside the cells, especially some of the antioxidants, then glutathione, they are being depleted or the lipid peroxidation is happening. In this cascade of events, it also causes ROS generation inside the mitochondria. Now ROS or the reactive oxygen species, it can be of several types. It can be hydroxide, it can be peroxide, it can be superoxide. So it's very important to understand what is the identity of the ROS that is being generated inside the cells. So based on this mitosox experiment, we found that it's the superoxide radical which is being generated. And we saw the effect of the mitochondrial membrane potential also with these assays. Now, all these, as I said, events, the downstream these events is that eventually we see endoplasmic reticulum stress is being. So endoplasmic reticulum, it is one of the major site which leads to unfolded protein response cascade to get activated. So what we started with? We started with where we want to convert the cold tumor into hot tumors. That was our initial goal, right? So eventually that's what we want to do with these class of compounds. So here this unfolded protein response cascade gets activated over here. There are two major proteins which plays very significant role to upregulate this cascade. One is the BIP protein and the other one is the PERK protein. So both these proteins, they act as a chaperone for the misfolded protein to be converted into folded proteins. So we saw that PERK protein, it's getting down-regulated, whereas BIP protein, it is getting up-regulated, which is one of the marker for the endoplasmic reticulum stress being activated. Now, coming back to ground zero, in order to show that whether you are converting the cold tumor into hot tumors, there were three major markers that you eventually want to see. The first one was this HMGB1 release, the second one is this ATP release, and the third one is this calreticulate. And we did see by immunofluorescence that all three of them does get affected. If you see this HMGB protein, it is going down, so immunofluorescence or the green fluorescence around these cancer cells, it's depleting over the period of time. The ATP, cellular ATP, it is being released, which is the find me cells, it is coming out. And the third one is the calreticulate protein, which is getting translocated from the endoplasmic reticulum to the cell surface, and we do see the increase in this green fluorescence over here. So we started with class of the compounds, ligands, we made their transition metal complexes, we characterized them, and eventually through the cascade of different experiments, we eventually proved that our compounds, they are participating in the conversion of the cold tumors into hot tumors. But so far all the experiments that I have shown you, they were done in the 96 well plates, they were done in the 2D system, okay. But if you think about the actual tumors, they are in the form of 3D spheroids, right? So we do 3D spheroids, especially, like in those 96 well plates, but still they are not very translational, I would say. So that's when we move to show them in the in vivo setup, where we do it in the biopsy mice. It's a 40 tumor one. So here we inject or inoculate. Yeah, so here we inoculate the tumor into the biopsy mice, and once the inoculation is done, once the tumor is established, then we do the intravenous injection for our compounds. After the intravenous injection, we do several dosage of 10 milligram from day 1 to day 7, and on the 8th day, and during this time period, we do follow up what is the tumor size, what is the tumor volume, how is the weight, if there is any side effect on the mice, if there is any, something that should not be because many times you do the experiment in the in vitro setup. The moment you go from in vitro setup to the mice models, you will see that the effects are drastically different than what you are expecting. Same happens when you go from the mice model to actual patients. You are expecting that, okay, everything is going fine, everything is working, but when you go to the patient samples, the things are quite not in your favor. So we did see that these class of compounds, they are showing decrease in the tumor volume, and tumor weight was also decreasing, and during the process there was no side effects. We did not see any side effects on the blood cell morphology or the tissue morphology, and we also wanted to see what is the distribution in different organ. We did see some accumulation inside the heart, but one of the reason that we gave it, like we hypothesized about it, that most probably it's because of more concentration of mitochondria. That's why we are seeing more accumulation. Now, one interesting thing over here is that during the process, if you see, this is the control where our iron complex is not being treated, and this is the iron box which we synthesized, so that is treated. And if you see the amount of the lymphocyte compared to the control, so lymphocytes were the cells which we want to be recruited if we want the immune induction to be there. Okay, so these lymphocytes, their recruitment has happened, indicating that we have been successfully able to convert the cold. But two limitations. One was ICT induction.
Conclude. Your time is going to be up.
Yeah, okay. More minutes. So these class of compounds, they do participate in photoacoustic signals. So this study was done with Dr. Sanhita Sinha Roy here in IISc Bangalore, and these class of compounds, they also participate in the near IR2 signal induction. So where are we heading? Eventually, we want to have immortalized PDF cell lines and tumor organoids, so that there is a more personalized treatment. We can have organ on the chips, which can be, provided to people who want to screen their compounds. And with this, I would like to thank my past and the present group members, Indian Academy of Science, for their associateship. I'm also visiting professor at ENS, and thank you ACS and RSC for awards over the period of time, and last but not the least, funding agency for their generous grants and funding. Thank you, and I would be happy to take any questions.
Thank you, Dr. Ritika, for an excellent presentation. So I think, sum it up, what I understood is the glutathione thing. That's a very important molecule now, because people are taking it for anti-aging, and you really anti-age. If you look at Safali Jariwala's episode, it reduces your age to 42. So that's one thing, the molecules are very fantastic molecules. And it's an excellent piece of work. I enjoyed listening to you, and I open the talk for some questions. One or two questions, because...
Yes.
Yeah.
Yeah.
You said cold tumor and hot tumor.
Yes.
Suppose somebody has a colon cancer. Do half the people have cold tumor and half or is it tumor dependent?
So there are markers which are available. So in case of, it's definitely tumor dependent. But as I said, in some cases immunotherapy does give very good response. In the other cases,
my question is, suppose colon cancer, can you say it's a cold tumor?
No, you cannot.
So half. That's what
it will vary from patient to patient.
Okay,
We cannot.
that's what I want. Thank you.
Okay.
Yeah, please.
Just one question.
Yes.
You talked about the involvement of unfolded protein response.
Yes.
So you have looked at the POD branch. Have you looked at, do you have any idea whether the other branches like IIT alpha, 1 alpha or ATF6 has any role in this?
Yes. So, yes, you are correct that, as I said, since we are not molecular biologists, so that's very diverse field of study that needs to be done. And people who are molecular biologists, they do study in lot more markers than what I have shown, even for the UPR cascade induction. So no, we did not see, but that's something definitely over the period of time we will try to include in our study.
So very nice presentation.
Thank you.
So I want to ask one thing, because you showed that in the last the CBC shows neutrophils and all those markers upregulated, but they may be upregulated because of the material you are giving. They are producing response against the material rather than your cold cancer cells is converting into hot cancer cell.
Absolutely.
So what is your comment on it?
Absolutely, you are absolutely correct that it might be because of inflammation, it might be because we are giving something external foreign, right? What we wanted to show that, it's actually a combined effect that we are seeing along with the markers, along with recruitment of the lymphocytes. But what you are saying, it's absolutely possible that it might be because of the compounds that we are getting, it's getting inflamed and the lymphocytes and all are increasing.
Ritika, you have shown a complex FeL1. So
Yes,
is
sir.
it soluble in water?
Yes, it is. But most of the time, one of the major limitation with our study is some compounds, they are not soluble. So
What
in
about
that
this
case, we try,
FeL1?
what is the FeL1?
No, you have shown.
Sorry.
You have shown.
Ah.
Yeah, you have shown in your slide
Yes.
that the complex which you synthesized that you have indicated to be FeL1.
Yeah.
So it is having many aromatic rings. So these aromatic rings may render its non-soluble properties in water. So it is soluble or insoluble?
This one is soluble, but what happens in case if it is not soluble, we try to dissolve it in DMSO, but there is also condition with DMSO that there has to be this many, like 0.1% or less than 0.1%, so that it doesn't become toxic. So in that cases, we try to do different formulation to increase the solubility.
Hi, very nice presentation.
Thank you.
But most of your screening you are done in human cell lines. How you correlate with your in vivo, which is a syngenic model?
Yes. So what happened when we moved from our human cancer cell line to the in vivo model? So first we went for the immunocompromised non-apoptotic mice. And there we tried injecting the same cancer cell line, the human cancer cell line. But first of all, they were very expensive. We are just into 4 or 5 years of our independent career. So just 8 to 9 mice were more than lakhs. So we did not get the tumor formation in those class of the mice. And then also keeping those mice having the optimum condition for those immunocompromised mice, it's actually like they will die out if we do not have those sterile conditions. So that's why at this stage we did 41, but eventually we are collaborating with the groups which can provide us immunocompromised mice, and we
because this MDM331, they are slow growing mice.
Yeah.
Better we can go with NSG mice so that you can get a better xenograph models.
Yes.
Let's thank Dr. Ritika with an applause.
Thank you.